Hello, this is my data analysis project. So my main takeaway from my project was that there was a statistically significant difference in photosynthetic activity between untreated corals and corals that had been in indirect contact with sponges. This treatment effect was similar across two different corals and sponge populations on the planet. So these are the two graphs that I created with my data, and they're both comparing control groups with a treatment group, and it's measuring their photosynthetic activity. On the left, it's measuring the maximum photosynthetic capacity, and on the right, it's measuring the maximum photosynthetic quantum yield. And so, after taking out this data, finding the averages, I was able to calculate standard errors and standard deviations so that I could look at the distribution of the data. I also calculated the t-value, showing that there was a statistical difference between the control group and the treated group. So both of the treated groups were treated very similarly. They were in indirect contact with sponges, allowing for the secondary metabolites of the sponges to be emitted to the corals, and then it was measured. Their photosynthetic activity was measured after the fact. And so as you can tell, there are different corals, different sponges, and also from different locations. So this was the figure that I came up with my two data sets, and I wanted to basically catch comparisons between the treatment effects, see if their mean outcomes between treated and untreated corals were similar in any way, even though they were from two different spots on the planet. And I took a t-value, and it was very small, and it kind of told me that these treatment effects were very similar, and that even though they're measuring different kinds of photosynthetic activity, they both affected the corals similarly, being in direct contact with sponges. So why is this important? So it's important to note that sea sponges and corals are very important for the ecosystem. The sea sponge being an important part of the carbon-nitrogen cycle, and then the corals being home for many sea life and ecological interactions. And so part of a survivorship, sea sponges emit these allelochemicals in order to protect their occupied space. So with additional stressors such as climate change, human pollution, ocean acidification, overfishing, they're going to release these allelochemicals to reduce their stress and fight against competitors. And so it's important to study these interactions because you want to make sure you are keeping proper balance, and so that it doesn't lead to a coral bleaching event or overgrowth of sponges. So for my first paper, I took out this figure that measured the average fluxes of net photosynthesis over small increments of irradiance of 4 different types of corals with different treatments. And so I used WebPlot Digitizer, and I took the no contact curve as well as the sponge excited curve, and I was able to grab these data points. And from that, I calculated average value for each group as well as both groups together as a population. And I also took the average maximum photosynthetic capacity of the last 3 points so that I could see where this curve kind of balanced off and where they got most of their photosynthesis from. And I was able also to calculate standard deviations and errors so that I could look at the distribution of the data. And lastly, for my second paper, I pulled out this figure, and it showed the average photosynthetic quantum yield of untreated corals as well as several corals that had been treated with secondary metabolites. So in order to have comparable values for the treatment effect, I calculated the average values of the 4 corals that were treated with allelochemicals, and then I compared that with the control value. I also calculated standard deviations and errors to look at the distribution of the data. So allowing to have the averages from both populations allowed me to catch comparisons and the mean outcomes between treated and untreated corals, and find that there was a similar treatment effect for corals and sponges in this interaction.